And this goes also with muscle tone, okay? We have to have a certain stabilization of the joints. So muscles are going to pull somewhat on tendons to create the stability to hold the joints up. You're standing up, your knees do not buckle. Your head is erect up as opposed to it flopping down with your chin hitting your chest. I mean, just all these type of things is part of posture and part of the stability of the joints to just maintain a certain uh, structure of the body. Another thing that muscles do is generate heat. If you remember our first week of lecture, we talked about homeostasis. And if you think about that, what is homeostasis? When we talk about generating heat, we're going to have the shivering. Okay, I know some people are probably shivering lately because it's so cold. But what is shivering? It is an approach in which the brain, to help the body generate heat, creates rapid muscular contractions, particularly with the skeletal muscles now. And that purpose is not to move things, not to maintain posture or stabilize joints. It is to generate heat. So you have rapid muscle contractions that are essential to help the body maintain heat, maintain homeostasis in the midst of, let's say, a very low temperature environment. From here, we're going to start up into the gross anatomy. And with skeletal muscle, you can see this. Remember, gross anatomy versus the microanatomy. Gross anatomy is large view of large structures. Microanatomy would be, of course, a much more smaller anatomical analysis or structural analysis. And we're going to get into that in a little while. But first, you got to keep in mind that muscles are a discrete organ. What I mean by that, remember the definition of an organ. It is a structure that is composed of different tissues. So what are you going to find in this muscle? Connective tissue, blood vessels, nerves. You're going to also have a certain amount of the muscle cells. The connective tissue is really sheets of connective tissue that allow the transfer of the contractile forces from the muscle fibers to the outside skeleton. So in essence, if you think about it, why is a tendon going to be so dense and is going to be so essential in its function? It is actually conducting a lot of the contractile forces that have been elicited or created by each of those individual fibers, and they get summated in a way through the tendon to where the tendon is attached to the bone. Now, remember we talked about origin and insertion? Well, we'll come back to this in a little bit because it is important. But when you start looking at the connective tissue, it also will contribute to the elasticity of the muscle. So the amount of flex and recoil is extremely important. Now, to help you again with some vocabulary, the prefixes epi refers to outer. Peri refers to around. So if you notice for the connective tissues, we have an epimysium, okay? And then we have an endomysium. The epimysium basically encircles, is outside of the muscle. It's an overcoat of dense, irregular connective tissues surrounding the whole muscle. The endomysium is within the muscle. It is connective tissue, mostly reticular fibers, which surround individual muscle fibers. But wait, we have more. Yes, it sounds like an infomercial. We have something in between that because we have these bundles of muscle fibers called fascicles. And so we have a peri, which means around the muscle, of fibrous connective tissue that surrounds the fascicles. Now, inside of a fascicle are individual muscle fibers. So then it's almost like if you've ever seen those Russian dolls where a smaller one will go inside of a next size up and the next one size up will go into another one and another one and another one. We're going from the large muscle structure to what makes up that muscle structure is the fascicles. And what makes up the fascicles are bundles of muscle fibers. Each level has a layer of connective tissue, epimysium, perimysium, and endomysium that is basically wrapping around certain structures. Okay, I hope that helps you there. And you can see this in this diagram. Nerves. Well, nerves are basically the skeletal muscle is dependent on nerve stimulation. That's one thing you have to understand. Nerves have to be triggered and to send signals to get the muscles to contract. We're going to get much more into this later on. You're going to hear about end plates and you're going to hear about neuromuscular junctions, and this is extremely important. You have to think about this from the perspective of consider the muscle without a nerve stimulation. And that can happen. If you think about this, somebody severs a spinal cord, what happens to all the muscles downstream? If they're not receiving any type of stimulation, they're going to start to deteriorate. And we'll get into this in a little while. Now, muscles consume a lot of energy. And as a result, they're going to require a rich blood supply to deliver oxygen and nutrients, as well as to remove waste. And keep that in the back of your mind because removal of waste is going to be paramount to the length of the capacity of the muscle to contract and the rigor of which it may be able to do that. You have to keep in mind that blood capillaries adjust to accommodate changes in muscle fiber contractions. Now, from here, we get into origin and insertion. And if you remember, we talked about this a little bit ago when we talked about joints. I wanted to get you introduced into it early, and this way it'll help reinforce it. When we have a muscle attachment to a bone, if the muscle attachment site to the bone is at a bone where the bone does not move, this is referred to as the origin. For the muscle attachment site to a bone where the bone does move, this is referred to as the insertion. So let's take a look at biceps brachii. If you notice where the O's are in this diagram up here, you see basically the O's, and they're attached to several structures on the scapula. Okay? Now, that scapula does not move. Think about it this way when you move your arm up. Okay? The forearm, the lower part will move. You have the long head and the short head of the biceps brachii. The origin site is not moving. The insertion site, where basically you have the radius and the ulna, where the tendon on the other side of that biceps brachii, they're attached. So when you have contraction of the biceps brachii, those bones move. Therefore, it's the insertion. Okay? Now, we have to keep in mind that there are different muscle attachment formats. We have the direct, which is referred to as fleshy. That's where the epimysium is fused directly to the bone or to cartilage. Okay? Indirect, the epimysium extends beyond the muscle like a rope-like structure, which we would call a tendon, or it can be to a sheet-like format, which is called an aponeurosis. Now, these are connective tissues. You've got to understand this. So we'll take a look at this from the temporalis muscle. If you look at the temporalis muscle, which is right around here, you see where you have the upper part, you have a direct attachment. The epimysium is fused with the perosteum of the bone. But down at the bottom here, you have an indirect, and you have an aponeurosis right low here, right next to the ear. That's going to have an attachment to the zygomatic part of the skull. Now, if you think about this, here's another example. Take a look over here. You have the occipital belly, and you have the frontal, what they might call the frontal belly, and this is all making up the epicranius. Here in the center, you have this large flattened sheet of connective tissue. It's called the galea aponeurotica. Basically, it has a connection to the occipital belly as well as a connection to the frontal belly, the frontalis muscle, and the occipitalis muscle. All right? Now, I hope that makes it somewhat clear. I mean, it's relatively easy to go back here and say, well, look, this is the tendon here, and this is the tendon here of the biceps brachii. Here's a tendon here of the biceps brachii. Yeah, no problem. But then you have to think about it. Not all tendons are rope-like. They're flattened out sheets, and that's the aponeurotica, okay? Let's move away from this and start moving into the microscopic anatomy. And this is where you would see this in figure 9 too. You have to think about this again from a structural organizational levels. Keep in mind that we have talked about the sarcolemma, which is the muscle cell membrane, and the sarcoplasm, which is the muscle cell plasm. The cell diameter, it's about 10 to 100 micrometers. Remember, micrometer is the equivalent of micron. It's a millionth of a meter. Yet the length may be up to several hundred centimeters. So in other words, if you look at some of the large muscles in the quadriceps or in, for example, some of the larger muscles in the back or even the biceps brachii, some of those cells, they're very, very narrow, but they're very, very long. A lot of cells you'll find are multinucleate because this cell is referred to as a syncytium. Syncytium means fused cell. So in other words, the cell may undergo repeated mitosis and you have multiple nuclei there, but that doesn't interfere with the function of the cell. The cell is just not going to divide. It expands and continues and builds up more and more of the contractile elements. You will find also inside that cell lots of glycosomes. If you remember, glycosomes are glycogen granules. Remember what glycogen is. It's a long polysaccharide made up of continuous glucoses, and they all have basically a covalent bond attached to them. Think about it this way. Where you're going to have need for lots of fuel, this is the perfect situation. You've got glycogen granules there. They can very quickly be broken down to provide glucose, which is fuel for the contraction of the muscle. Myoglobin. Myoglobin provides the red pigment of the muscles. Myoglobin is similar to hemoglobin, except it's just one chain of protein, as opposed to hemoglobin, which is composed of 4 chains. But the myoglobin works very similar to hemoglobin. It holds onto oxygen, stores it, and then will release it when there's a need. You're also going to find lots and lots of those contractile elements inside of that muscle. That's evidence of the myofibrils. You'll see the banding there, the dark A band, which would be the striation. You have a light I band there, and that's the fading out. Also, what you'll find are present are T-tubules in the sarcoplasmic reticulum. The sarcoplasmic reticulum is really a modified smooth endoplasmic reticulum, and this is part of the signaling system, that occurs that will trigger the muscle contraction. One other thing that you're going to find lots and lots and lots of are mitochondria, because as we've talked before, the mitochondria are the cells, or excuse me, the organelles that basically power the cell. They are the powerhouse of the cell. They produce lots and lots of ATP, and you're going to need lots and lots of ATP, as you see, for the contraction of these myofibrils. When you look at the myofibril structure, basically what you have is about 80% of the cell volume is made up of 2 filaments, actin, myosin. Actin is referred to as the thin filament, myosin is the thick filament, okay? And you can see how that nucleus is pasted close up against and pushed tightly up against the sarcolemma. We're moving now into looking at the bands. Now, yes, you need to know these, okay? Hint, hint, it helps, okay? Now, you're going to find that there is a structure called the H zone. If you look at this diagram, and this is the middle line of the myosin thick filament. Basically, when we talk about a sarcomere, that is the length of one contractile unit from Z disc